introduction. Good morning, everyone. My name is Sunandita Sarker. I'm very excited to be back here today, and also this is my first conference after accepting my position as an assistant professor at UMass Amherst. Today, I will discuss about my recent work involving additive manufacturing to create novel biopsy tools that could benefit pediatrics with complex congenital heart diseases. Congenital heart disease, or CHDs, are structural heart defects present from birth. These conditions range from simple anomalies like small holes in the heart to complex malformations requiring advanced medical treatments and surgeries. For severe and end-stage CHD, a heart transplant is often the only viable option. According to the American Heart Association, about 12% of all heart transplants are pediatric cases. Post-transplant care includes frequent biopsies, especially patients typically require about 10 to 12 heart biopsies in the very first year of transplant. Heart biopsy, or endomyocardial biopsy, is a catheter-based process where the catheter is inserted through the vein, either a jugular or femoral vein, and guided to the heart using X-ray fluoroscopy. Small pieces of heart tissue are removed and analyzed for signs of rejections or infections. Even though this procedure is a crucial part of post-transplant care, it comes with several challenges, particularly for pediatric patients. First, these technologies are X-ray dependent, and they're quite large, typically in the range of 5 to 7 French, which is about 2 millimeters in outer diameter. Additionally, these instruments require manual bending, which limits their maneuverability to certain location of the heart, and it could also increase the chances of unintentional injuries. In recent years, researchers have been investigating soft robots for different surgical applications, including biopsies. While most work focuses on lung biopsies or gastrointestinal biopsies, their potential for cardiac intervention is also being explored. Soft-robotic tools offer controlled maneuverability that could help reach target locations, minimizing injuries. However, the current tools are also very large, especially in the millimeter to centimeter range. So their application in pediatric cases are limited due to the complex geometry and the smaller anatomy of a child. So the challenge is, how do we manufacture biopsy tools at a micro scale that could be suitable for pediatrics? So here, we're proposing that 3D microprinting, in particular 2-photon direct laser writing-based microprinting, can be used to create these micro-scale biopsy tools that could be suitable for pediatric endomyocardial biopsy. Previously, we demonstrated an ex situ direct laser writing approach, which involves 3D microprinting structures directly on top of larger scale capillaries or fluidic components. In this case, we showed a 3D microneedle array on top of capillaries. We validated our device using excised mouse brain tissue and demonstrated safe tissue penetration and retraction. We have also used esDLW to 3D microprint singular high aspect ratio microneedles that could be utilized for IVF or developmental biology research, and we have used these singular microneedles to perform serial microinjections on decorionated zebrafish embryos. So besides our microneedle work, we have utilized esDLW to build soft-robotic surgical guide wire tips directly on top of mesoscale tubings. The soft actuator of the guide wire is designed to form specific S-shaped geometries that could facilitate navigation. So today, I'm presenting an ex situ direct laser writing-based approach to manufacture a microrobotic biopsy tool with a soft actuator and a biopsy needle. Using a custom-made holder, the large-scale capillary can be loaded into 3D nanoprinter, and then a photocurable material can be deposited on top of the material inside the printer. Next, a pulsed infrared laser scans through the material, polymerizing only at designated location in a point-by-point, layer-by-layer manner to print the entire biopsy tool. Once pressure is applied at the inlet, the soft actuator can extend for tissue penetration. Here, the computer-aided manufacturing simulation and corresponding printing process video shows how the biopsy tool is fabricated directly on top of the capillary. We usually start the 3D microprinting process with a 100 micron overlap to make sure proper adhesion with the tubing. Here's the SEM micrograph of the biopsy tool onto the capillary. So initially, we performed 2 sets of proof of concept experiments to investigate the mechanical capabilities of the biopsy tool. So using a microstage, we first investigated the ability of the biopsy tool to penetrate 10 weight percent agarose gel. As shown, we did not observe any mechanical failure such as buckling or breaking of the tip. Then, we tested the ability of the biopsy tool to penetrate biological tissue ex vivo. We collected some excised bovine heart tissue for this puncture experiment. As shown in this video, our biopsy tool was successful in puncturing the tissue and then retract. Next, we investigated the capability of the soft-robotic actuator. We applied fluidic pressure at the inlet and gradually increased it to 600 kilopascal, while the biopsy tool maintained a very strong mechanical integrity. It exhibited very limited actuation under high pressure. So we investigated to improve the actuation capability using computational analysis. So we conducted FSI simulations where we used the material properties similar to the IP-PDMS photomaterial that we have been using to 3D print our biopsy tools. And despite analyzing various geometric variation, we observed only minor improvements with this material. So we then explored a different material, IP-PDMS, and this material is known for its flexibility. The simulation results demonstrated much larger actuation with IP-PDMS. However, a needle with IP-PDMS is too soft to puncture any tissue effectively. That led us to our second concept, a dual material concept, where we have the actuator 3D printed with the soft IP-PDMS material that will facilitate actuation, while the needle is printed with a much stiffer IP-PDMS material for better tissue puncture. During the print process, we first print the actuator with IP-PDMS, then we clear the material from the printer and replace it with IP-PDMS to 3D print the needle. And similarly, once pressure is applied at the inlet, we can observe actuation for both puncture and navigation. Here's the SEM micrograph of the dual material biopsy tool. And here you can see the interface between the two materials. So now we investigated the fluidic actuation capability, which was the motivation of this dual concept. And we first applied a vacuum pressure to retract the biopsy tool to the minimum length, and then we increased the pressure. This result indicates much more effective actuation compared to the single material biopsy tool. So it's still a work in progress, but to conclude today, in a summary, here we introduced the Situ direct laser writing-based 3D microprinting of soft-robotic biopsy tools. We demonstrated both single and dual material biopsy tools at a 1 French outer diameter, and to our knowledge, this is the first time a biopsy tool at this length scale. We also validated the mechanical capability of the biopsy tool both in phantoms and bovine heart tissues. Moving forward, we are investigating the ability to puncture different stiffness phantoms and tissues using the IP-PDMS soft actuator mechanism. We are also interested in exploring robotic maneuverability for navigation to target location using our IP-PDMS soft actuator system. I would like to acknowledge all the funding agencies for supporting the different works presented in today's talk. I would also like to thank UMD Terpin Works and Princeton Materials Institute for Micro and Nanofabrication Center for their cooperation. Lastly, I would like to thank my co-authors and everyone in the BAM lab for their contributions. Especially, I would like to extend my gratitude to my mentor, Dr. Ryan Sokol, for his unwavering support. As much as I will miss my time at University of Maryland, I'm thrilled to announce that I'm launching my own lab, Technologies for Healthcare Enhancement via Advanced Manufacturing, or THEME Lab, at UMass Amherst this fall. Here I will address various design and manufacturing challenges for biomedical applications. I'm eager to collaborate and engage more with our Hilton Head community, so please do not hesitate to contact me. With that, I would like to thank you all, and now I open the floor for questions.